students here who have made my visit so fabulous, and just for showing me a great time and sharing your science with me. I really had an amazing visit. And what I thought I wanted to do tonight is to tell you a story about CRISPR. I want to start with how I got involved in this field, which until not too long ago wasn't even a field. And then I really want to share with you where this is going as a technology, what's happening today, what's going to be happening in the not too distant future, and some of the really exciting things that I think are going to happen with gene editing, but also some of the important challenges that we face both ethically and societally, thinking about how we use a powerful tool for altering genomes in ways that are going to enhance and help human life and human societies without taking us places that we don't want to go with this technology. And so just to get us going, there's my title. And I wanted to start by just pointing out that, especially for students here, this project, for me, started as a curiosity-driven set of experiments that led in an unexpected direction. It's kind of why we do science, right? We want to satisfy our curiosity about nature, about the world, and every now and then things happen that you couldn't have predicted that take your work to a place that you couldn't have imagined. And that's absolutely what happened to me and to my lab. And so we started studying how bacteria fight viral infection. This is a slide that shows a photograph of a bacterium that's being attacked by viruses, and they use different ways of defending themselves against viruses. And in the course of investigating how this works for a particular type of pathway, we came across an enzyme that can be used to alter genomes. And I'm going to explain what that is, and then we'll talk about a little bit about how it works and where it's going. So for me, my whole awareness of what's known as CRISPR biology came about back in around, I think it was 2006 or so, when a colleague of mine at Berkeley, Jill Banfield, told me about something mysterious in her own research. So she is a scientist who works on bacteria. She studies bacteria that are found in various environmental niches, and her work involves sequencing their DNA to find out what kinds of microbes are inhabiting our planet. And in the course of her work, she discovered that many bacteria have an unusual sequence in their genome, in their DNA, that includes a repetitive sequence element that flanks unique bits of sequence that in around 2005 were found to derive from viruses. And in fact, they came from the viruses that infect these bacteria. So this was the first hint that these unusual DNA elements were somehow representing a bacterial immune system, a way that bacteria could store genetic information and then maybe use it to protect cells in the future. This is a slide, this actually shows a cartoon of the circular chromosome of a bacterium, and here's the element that we call CRISPR, and it includes these bits of DNA sequence that are each unique, and they come from viruses. And importantly, these sequences are found usually right next to CRISPR-associated or Cas genes. These are genes that encode proteins that initially, again, were of unknown function, but were always found next to these CRISPR arrays. And so that led to the idea that maybe bacteria have an adaptive immune system that operates by an unknown mechanism, and that's where our lab got involved. So what emerged over the few years after my initial conversation with Jill Banfield, and it was largely from research going on in just a handful of labs around the world, and that included at a yogurt company. It included scientists who were working in the dairy industry. These were people that were studying microbes that can be infectious in food and how to try to protect food cultures from these bugs. And so they were very interested in how bacteria might fight off viruses. And they discovered that in bacteria that have these CRISPR systems, the cells can literally detect a viral infection and integrate DNA that would be injected from a virus. So here's the cartoon showing a virus landing on the surface of a bacterium, injecting its DNA. And if the cell has a CRISPR system in the genome, it can integrate a piece of the viral sequence into the array. And And then these sequences, and this is where my lab's expertise came in, these sequences operate not at the level of DNA, but they actually function as RNA molecules that are made, they're transcribed in the cell. So the RNA is a copy of that CRISPR array, and each of the molecules of RNA that result from the cleavage of this initial piece of RNA that's generated provide a single sequence that can recognize a matching sequence of DNA. And these RNAs don't work alone, they actually combine with one or more CRISPR Cas proteins, the proteins encoded by the Cas genes, to form RNA-guided protein complexes. They can surveil the cell looking for matching DNA sequences, and when they find a match, the Cas proteins are recruited to that piece of DNA and they cut it up. So it's a fantastic way that bacteria can adapt to viruses and provide a memory of infection, and not only that, use that memory to protect the cell from future infection. So the biology was very cool, right? It was really fun to kind of figure this out, and which was not done by my lab, by the way. And but where we came in was asking how. How does that work? We wanted to understand the molecules that actually operate in this pathway, how they function, and how they provide this kind of protection. And I've always been fascinated also by evolution. Where did these come from? I wanted to understand, how is it that bacteria have evolved this system? Why don't we see it in us, for example, which we don't? And so how do we understand that? And so the question was where to get started. And so, as I mentioned, my colleague Jill Banfield at Berkeley investigates biology by looking at bacterial organisms that are growing in the environment, and her research involves literally going out and collecting samples. And this is a slide showing how they do this sampling. So these are students that are out in the environment. They're out, in this case, in Northern California, and they're collecting water and soil samples. And then they can see on the left-hand side, they've got this sort of plumb line where they've got all of these little tubes that are collecting different samples. They can mark where they come from. And then these samples are taken back to the laboratory, and you can isolate bacteria from these samples and sequence their DNA. And that's exactly what they did. And what came out of all of that work, in addition to lots and lots of other data, was the discovery that there are many different kinds of CRISPR Cas systems, many kinds of these immune systems in bacteria. And this is a slide that just illustrates the different collections of proteins that are found in different CRISPR systems in bacteria. So each of these colored boxes represents a gene in the genome that encodes a Cas protein. And they're organized according to types. So the three types at the top that belong to class 1 are all systems that require many, many proteins. You can see many, many colored boxes there. Many proteins to protect cells, compared to what you see on the bottom, these class 2 systems that typically have one very large gene that is involved in protecting the cells from future infection. And so it was in the course of our research, we started off investigating one type of CRISPR Cas system, and then I went to a conference and met a colleague, Emmanuel Charpentier, who works on basically microbes that infect human beings. And she had been working on a bug that had one of these microbes that contains a different kind of CRISPR system than my lab had been studying up until that point. This one right here, type 2, that includes a single large gene known as Cas9, responsible for the function of this immune system in that bacterium. And so our meeting led to a question that we wanted to address together as collaborators to figure out what was the function of CRISPR-Cas9. And I still remember having this conversation with her. We were in Puerto Rico at this conference and talking about what our labs were each doing, and we realized that we had complementary skill sets. I'm a biochemist and a structural biologist, so my lab has always worked on molecules, and RNA has been at the heart of our work in many ways. Her lab studies bacteria that infect humans and tries to understand the biology of bacteria from the perspective of how they interact with human hosts. And so we were each coming at this CRISPR pathway from different perspectives, but we were fascinated by this question. And what happened next was truly extraordinary, because we went back to our laboratories, hers in Sweden, mine in Berkeley, And we engaged our students, in my lab, Martin Jinek, in her lab, Chris Chylinski, to investigate this question. And so the way we attacked it, the way we went about it, was to start purifying the Cas9 protein to test its biochemical activity. And what Martin and Chris in our labs figured out was that this protein Cas9 is an RNA-guided enzyme, and it works by using the CRISPR RNA molecule, which is shown in this cartoon right here, with its 20-letter sequence that recognizes DNA. It uses that interaction to direct the Cas9 protein to a particular place in a molecule of DNA. And that molecule could be very tiny, or it could be an entire chromosome. And when it finds that position, it allows the Cas9 protein to unwind the DNA helix and generate a double-stranded DNA break in the DNA. So that was pretty cool. And then Martin and Chris figured out that in nature, this system requires two types of RNA. Not only this CRISPR molecule that does provides the address label for the protein, but also this second kind of RNA called tracer that is essential for forming a structure that can associate with Cas9. So it's a dual RNA-guided system. And as Martin Jinek was playing around with this and testing what was minimally required for this DNA cutting reaction to work, he figured out that the two RNAs could actually be linked together in what we call a single guide RNA, a single molecule of RNA that would contain the address label for recognizing DNA and also the essential structure that would be necessary for binding to Cas9. And he did a great experiment. I told the students about this today. It was the simplest experiment, for a molecular biologist that you can sort of imagine, where you take this single guide RNA, and Martin made 5 different versions of it that would recognize different sequences in DNA, and then we just incubated it with Cas9 and a plasmid DNA molecule. This is a circular piece of DNA purified that we could test in the laboratory. And we saw very cleanly that the Cas9 protein would cut the plasmid at precisely the position that it was programmed to cut. And it was one of those very, very simple but very kind of beautiful results that, in my case, changed my life, because we saw this result and we realized that our project that had started as a curiosity-driven question about the function of Cas9 was going in a very different and very, very exciting direction, because we realized that this chemical cleaver of DNA, which could be programmed to cut DNA at a desired place, could actually be utilized to do something very different in human and animal and plant cells, namely to trigger genome editing, changes to the genome at a precise position. And to explain that, I have to show you another cartoon that illustrates what happens in our cells, plants and animal cells, when the DNA experiences a double-stranded break. So unlike in bacteria, where double-stranded DNA breaks in DNA lead to rapid destruction of the DNA, so that's a great mechanism for getting rid of viruses in the case of this CRISPR system. In our cells and in plants and animals, when a double-stranded break occurs, the cell detects the break and fixes it. And in the process of fixing it, one can introduce a change to the DNA sequence. And that change can be small, like shown on the left of this slide, or the change can be much bigger. In fact, it can result in incorporation of new genetic information at the site of the break. And so many scientists had recognized this over the last couple of decades and had been working on technologies for introducing a double-stranded DNA break at a desired place. And that was the challenge, right? How to do that. And there were earlier technologies that worked very well, but were cumbersome to use. It was hard to implement them because they required a lot of protein engineering. You had to be an expert in working with and engineering proteins to bind particular places in DNA. And when we uncovered the activity of this Cas9 enzyme, we realized that nature had already figured this out. It had already figured out how you could program a single enzyme to cut DNA in the desired place, and that as scientists we could now control the activity of that protein because we knew how its programming worked. And so I want to show you a little video that illustrates how we imagine that this bacterial enzyme operates in a eukaryotic cell. So here we are, we're zooming into a cell. So in animal and plant cells, the DNA is inside the nucleus, And not only that, it's packaged as chromatin, so the blue DNA is wound around green histone protein, so it's highly compacted. And amazingly, what Cas9 does is it searches through the cell looking for a place in the DNA that matches the sequence of the guide RNA that it contains. When that match is found, it unwinds the DNA and opens up the helix, allows the Cas9 protein to cut the DNA, and then these broken ends of DNA are handed off to repair enzymes in the cell that take over and fix the break, like in this example, actually integrating a new piece of sequence in the course of repairing the break. And as soon as we published this work in the summer of 2012, labs around the world began to investigate the use of this technology in all sorts of systems. And within 6 months, there were publications coming out from several different labs that showed that you could use this CRISPR-Cas9 gene editing tool in human cells, but also in plant cells, even in whole organisms like zebrafish, to make changes to the genome in targeted ways.